I'm Hannah Donnert with the Collaborative on Health and the Environment. Change is bringing you the latest environmental health science through our partnerships, calls, webinars, science serves, publications, and social media. I would like to welcome everyone to today's webinar entitled Mechanistic Mediation of Flame Retardants in Preterm Birth. This webinar is sponsored by the CHE-EDC Strategies Partnership, which is chaired by Cheryl Patton, Director of Commonwealth's Biomonitoring Resource Center, Jerry Heindel, Founder and Director of Commonwealth Healthy Environment and Endocrine Disruptor Strategies, Janan Jensen, Executive Director of Health and Environment Alliance, Sarah Howard, Founder of DiabetesEnvironment.org, and the Collaborative on Health and the Environment, yours truly. Our moderator today is Jerry Heindel, Founder and Director of Commonwealth's Health and Environment and Endocrine Disruptor Strategies, or HEEDS. We will have time following the presentations for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentations. After the presentation, our moderator will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who have called in on the phone, we posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speaker. This webinar is scheduled to last for 30 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Jerry.
Okay, thank you very much, and welcome everyone to our webinar today. As you heard, our speaker today is Dr. Ramkumar Menon. Dr. Menon is a tenured professor in the Department of Obstetrics and Gynecology and Cell Biology at the University of Texas Medical Branch in Galveston. He's also the director of the Division of Basic and Translational Research, and he's a global leader in preterm birth research and functions as executive director of the Preterm Birth International Collaborative, which is a not-for-profit research and education organization. His lab is developing strategies to reduce preterm birth using extracellular vesicles as delivery vehicles to treat fetal inflammatory responses. He is also involved in developing organ-on-a-chip models. He's published over 275 reviewed articles and book chapters, and he's also a mentor to over 50 medical and graduate students. As you'll hear today, his research also focuses on endocrine disruptors, specifically flame retardants and their role in preterm birth. So as I mentioned, while Dr. Menon is well known in the preterm birth, exosome, and organ-on-a-chip scientific communities, he's less well known in the endocrine disruptor community. So we believe this is very important that the endocrine disruptor community know more about the research he's doing. So we're very pleased to have him with us today to talk about his research. So welcome, Dr. Menon, and I give the floor to you.
Thank you. Thank you, Dr. Heindel and Hannah, for inviting me for this exciting opportunity to meet with the endocrine disruptor group of people that I've never heard about, actually, about this seminar space series, and I'm really honored to be a part of this this event, and for giving me the opportunity. Thank you so much for giving me the opportunity to talk about our research. And although I did not know when I signed up for this particular day, today is the World Prematurity Day. So it is such a privilege to talk about prevention of preterm birth, strategies of preterm birth, prevention of preterm birth, as well as causality of preterm birth on a day that is incredibly important to our research community. So I'm a reproductive biologist by training, and as a reproductive biologist, whether you are a scientist, whether you are a clinician, or whether you are a nurse practitioner or a nurse, the goal of us is to have a happy and a healthy pregnancy at the end of a 10-month pregnancy period. But unfortunately, not all pregnancies end as happy and healthy as prematurity or preterm birth. That complicates many of these pregnancies. As you can see on the slide, preterm birth is defined by WHO as a live birth before 37 weeks of complete gestation. As you very well may know, a normal birth, a normal term birth is anywhere at 40 weeks of gestation. 40 weeks is a complete mature birth, term birth. But anything that happens before that is preterm birth. There are 15 million preterm babies born each year, and 1 million of them will not see their first birthday as because of complications of prematurity caused to neonatal death. Now, if they survive, they will have lifelong disabilities and also experience adult onset diseases such as childhood asthma, diabetes, cardiovascular diseases, several forms of cancers because of complications of prematurity. So my job is to understand preterm birth, what causes it, how mechanistically several risk factors manifest it, and how we can prevent preterm birth from happening. So this is a very detailed figure, as you can see. If you look at the innermost circle here, the preterm labor, if you're a clinician working in the area, you're getting a patient that with preterm labor at the very end stage or terminal stage when there is no going back. You can only delay preterm delivery, but you are not going to stop it completely. So what happens is that these are the pathways: oxidative stress, inflammation. These are pathophysiological mechanisms that can actually trigger a series of events: placental senescence or aging, apoptosis, decidual activation. Decidua is one of the maternal tissue that has got several immune components get activated. Fetal membrane or placental membrane weakens and causes rupture. Myometrium, which is the muscle bag or the uterus, start to cause activation, which is called myometrium activation, is called labor. And cervix, the birth canal, start to ripen and opens up for delivery. So these are the events that leads to preterm labor. That actually leads to normal term birth too, in the absence of any of the risk factors. These are the same mechanism that actually leads to labor at term as well. But when it comes to preterm labor, you can see the circle outside of that. I have listed several other diseases that that can lead to these mechanistic event that leads to preterm labor. It can be allergies, infection, autoimmune disorders, uterine disorders, hydramnios, too much of amniotic fluid or too little amniotic fluid, hemorrhage in the decidua or endometrium, rupture of the membrane, preterm premature rupture of the membranes, and commonly called as breaking the water bag. That's all of these can lead to this pathway causing preterm labor. Endocrine dysfunction, I highlighted that because the group here are much more interested in that. That is one of the major factors associated with activation of this pathway and preterm labor. Now, these are dynamic risk factors. That means they develop during the course of pregnancy. But there are several static risk factors that is listed in the outermost circle here: genetic factors, ethnicity and race, nutritional, behavioral factors, psychosocial factors, socioeconomic factors, body mass index. These are more like a static risk factors because highly unlikely in many of the patients, these are not going to change during the course of pregnancy. And of course, the environment they live in. It's also the materials they get exposed to. They're not going to change in many course of pregnancy because you're not going to relocate just because you're pregnant. So these are static risk factors, and these static risk factors can impact epigenetically on maternal as well as fetal uterine tissue and can manifest as one of these diseases. And one of these diseases can cause the pathway, activate the pathway leading to preterm labor. So preterm birth is a very complex syndrome. When a clinician looks at preterm labor, they have no idea what risk factor actually triggered the mechanism that leads to preterm labor. So today I'm going to focus on environment. So here's an interesting thing that the room behind me actually is getting renovated, and the construction workers covered that up with plastic material. And I noticed that every one of those plastic sheet has a seal on them, flame retardant. it was kind of coincident that this work started about 2 weeks ago when Jerry actually asked me to put this presentation together about flame retardant. So then I asked these construction workers, what is this about? Oh yeah, we spray materials here, so that there's no fire hazard here. So that was kind of interesting. So how now I am exposed to this flame retardant on a daily basis because this is just outside of my door. So what is flame retardant? So what is PBDE, or polybromated diphenyl ether? It's one of the most common flame retardant used or sprayed on most of all surfaces that we touch, we inhale, we experience that on a daily basis. And the PBDE is one of the most prevalent organic pollutants in the world. And it actually is it's applied to numerous consumer products over the past 40 years. And PBDEs function, as I mentioned earlier, as a flame retardant. And normally they exist in different congeners. They are structurally similar, contain central biphenyl structure surrounded by up to 10 bromine atoms. And so based on the structure, so I'm not a biochemist, I'm not a chemist. So I don't know much about PBDE chemistry, and I'm sure that many of you in the audience actually listening to this talk may know much better than I do. I only look at functionally how it impacts pregnancy and what is the risk of pregnancy if a pregnant woman get exposed to PBDE during the course of her pregnancy. So we are specifically interested in PBDE congener 47 and PBDE-99. We have looked at many other congeners as well, but many of my slides today will focus on these two congeners. So when we initially started looking at PBDE's impact on preterm birth or birth before 37 weeks, we noticed that accumulation of PBDE, either in the amniotic fluid or maternal plasma sample or cord blood samples, are actually associated with risk of preterm birth. So depending, as you can see on the slide, I mean here the PBDE levels, and on this one, depending on the quantity of PBDE in maternal plasma samples, you can see the significance of risk that increases as the concentration of PBDE increases in maternal plasma. So these work are all published, and based on this, my interest is, so most of these are epidemiological studies that we have done using biobank samples from different pregnancy cohorts. This particular cohort that I'm showing data from came from Nashville, Tennessee, pregnant women in Nashville, Tennessee. So my interest is, okay, so it is increasing. It is most likely that it's unavoidable that a pregnant woman do not come in contact with PBDE. That's almost impossible. But if we understand how PBDE at this dose can manifest preterm birth, if we can understand the pathways of preterm birth, maybe we can provide some interventions. So that's my job. And if you look at what leads to preterm birth in general, normally, as I said, even if it is a normal birth, there are several signals that comes from the fetus as the fetus gets mature or the fetus has grown. It leads to an increased inflammation and oxidative stress in the intrauterine cavity or in the amniotic fluid, leads to cervical ripening, myometrial activation or uterine activation, which is called, and contractions, which is called labor, leading to delivery. But in preterm birth, so these in a green box, that normally happens around 40 weeks of pregnancy, which is normal. In some cases, almost 12% of women, it happens pathologically because of some form of risk exposure they will have during the course of pregnancy. It could be PBDE, it could be infection, it could be something else, it could be behavioral. And the same pathway is triggered in response to risk factors prematurely or earlier than 37 weeks of gestation that leads to preterm delivery rather than normal term delivery. Now, can we intervene? Most of the interventions are designed to stop cervical ripening or myometrial activation, which is maternal tissues that are maternal uterine tissues. We try to stop them from ripening or contracting, but it is way too late. You can delay delivery for a few days, but you cannot do it beyond that. So the actual cost, the inflammation and oxidative stress that is building up in the fetus is not necessarily addressed. So that's my lab's job. We look at fetus as a patient. and how the fetus triggers the mechanisms of parturition. And you can see in this figure, there are two interfaces that the fetus come in contact with the mother. One is the placental decidual interface, as you can see here, and the other one is fetal membrane decidual interface, placental decidual, fetal membrane decidual interface. And my lab is specifically interested in fetal membrane that is very close. This is basically the sac, amniotic sac, that where the fetus is held together, which is filled with amniotic fluid. So it's like the covering of a balloon. And this is the structure of amniotic membrane, amniochorionic membrane, or fetal membrane. This external layer, epithelial layer, is in close contact with the amniotic fluid where the baby is. And then there is an extracellular matrix. Then there is chorion trophoblast layer. This layer is in touch with maternal tissues, decidua and myometrium. So that forms the choriodecidual interface or feto-maternal interface. So my lab's interest is studying how this particular tissue, this is a fetal in origin tissue, and how this tissue responds to various chemicals or infectious agents. So we collected this amniotic epithelium. So what this is normally done in our lab. We isolate amniotic epithelial cells from normal term deliveries and grow them in culture. We examine what their markers are. So these are normally cytokeratin positive cells. We expose them to oxidative stress agent. And in this case, not oxidative stress agent, any other risk factor. In this case, we expose them to PBDE. Two of the congeners were tested, PBDE-99 and PBDE-47. And we saw that in these cells, if these PBDEs are in the amniotic fluid, it will expose these cells to those PBDE, and it will cause reactive oxygen species development. So you can see that PBDE-99 substantially increases the ROS radicals in these cells, not so much by 47 compared to control, which is here. And if you treat them with N-acetylcysteine, which is an antioxidant, you can see that both of these levels come back substantially, suggesting that PBDE-99 and 47 can induce oxidative stress and reactive oxygen species. So this is another experiment that we have shown in the past, that if these amnion epithelial cells or chorion trophoblast cells get exposed to oxidative stress, as shown in the panel on the left here, and this is a control, and you can see the damage that can cause to the epithelial layer. This is normal, this is oxidative stress. Look at the nuclei structure. Nuclei is distorted. Endoplasmic reticulum is actually bulked up. It's more like a ballooning appearance here. Same thing, mitochondria, whereas in normal, it is wormy looking, normal looking ER, normal looking mitochondria, dark mitochondria. All their morphology is twisted and changed in response to oxidative stress. So reactive oxygen species exposure of development in the amniotic cells can cause this morphological changes, which is basically damage to that cell. All organelles are impacted. Not only organelles are impacted, it causes DNA damage. And this DNA damage can trigger a pathway as shown in this figure. And we were specific, and what happens is it can impact the cell cycle progression. So if it is impacted in a unique way, it can either lead to cancer or it can lead to senescence or apoptosis, depending on how this pathway turns out. And again, this is something that we were interested in looking at. There are many other pathways that can also be triggered by DNA damage. And if the DNA damage or repair fails to happen, you can see that this kind of thing's happening. And we were more interested in senescence during pregnancy rather than cancer, because it is an end stage that is expected in 10 months. So what we are seeing is, in this particular case, we saw that 47, we looked at 47 and PBDE-99. This PBDE-99 at 48 hours and 72 hours caused phosphorylated P38 MAP kinase activation, not so much by 47. And here's the total P38. We did not see phosphorylation of P53 or even activation of P53. So what happens, so there is a significant increase at 72 hours that P38 went up in response to PBDE-99, and both 47 and 99 cause senescence-associated beta-galactosidase staining of amniotic cells, amniotic epithelial cells, compared to control, suggesting that these cells are undergoing senescence in response to PBDE. And what I'm not showing today is when we knocked out PBDE from these cells, we were able to minimize senescence in those cells. So PBDE-mediated senescence can be controlled by regulating P38 MAP kinase expression. And this senescence was also associated with a very unique set of cytokines and growth factors and other inflammatory mediators called SAS, senescence-associated secretory phenotype. And we saw many of these cytokines and growth factors going up in response to senescence compared to control, and basically in response to PBDE. And it also increased matrix degrading enzymes, MMP9, and prostaglandin, induced prostaglandin. So prostaglandin is one of the major inducers of labor. So that was also going up. So if you look at this, this figure, what we predicted is aging is happening in utero. Aging is not a late life phenomena. It actually starts in utero. If you look at development of the embryo, that's 2 cells in 2 days. And it undergo massive growth. And this at 30, and then it starts to, at term, when the fetus is completely mature, the fetus sends signals of senescence in the form of inflammation, and the fetal delivery happens. But if this is prematurely aging or senescing, then you can see that the birth can happen well before 37 weeks, leading to preterm birth. So in a normal situation, oxidative stress happens at 40 weeks. And these are some of the reasons why intrauterine cavity build up oxidative stress. And this leads to P38 activation, senescence. So this is a normal physiology. But in case of risk exposure by the mother, whether it is infection, stress, geographic location, BMI, nutrition, and environmental factors, or smoking, which is one of the major causes of major risk factor, it can activate the same pathway prematurely. So I'm going to talk a little bit more on the environmental side. Now, one of the things that we are interested, as Jerry mentioned, we are interested with how can a senescent cell trigger labor on the maternal side. We're looking at the fetal senescence, and we're talking about maternal activation. So our hypothesis, my lab's hypothesis, is that these inflammation or senescence-associated secretory phenotype gets packaged inside vesicles, extracellular vesicles, that can carry as signalers and to the mother and cause premature labor. So that's our hypothesis. It may happen normally in term labor. It may happen prematurely in preterm labor. So as you all may know, that extracellular vesicles are nano-sized particles at about 30 to 160 to 200 nanometers, released from almost all cells. They carry different types of cargoes, and these cargo, as proteins or nucleic acid or lipids or whatever, they can actually may have a functional impact when the exosomes are received by a different type of cell. So we hypothesized that fetal exosomes carrying inflammatory mediators from a PBDE exposed or any other environmental risk factor exposed tissue may carry inflammation to the maternal side, prematurely triggering activation of maternal uterine cavity. So we looked at PBDE exposed amniotic cell-derived exosomes. When we looked at the size of these exosomes, we looked at different congeners. We also looked at BPA, BPS, which I will not be getting into much into that today. But all the red bars are different congeners of PBDE. We did not see any change in the size of exosome released from the cells. We did not see a significant change in quantity of exosome released. We did not see any change in the shape of exosomes. So those two are shown electron micrographic images here. Didn't see any difference in size, shape, quantity, or even markers, which I haven't shown here. Exosome markers did not change in response to PBDE. However, the cargo was different compared to control. The cargo was different for different PBDE congeners. And you can see in this Venn diagram, there are several overlapping factors between exosome cargo, but there are several unique cargo contents also. So in one other example, one other experiments, when we exposed the cells to oxidative stress using cigarette smoke, one of the things that stood out in our bioinformatic analysis of these proteomic data was HMGB1 signaling. And as you very well may know, HMGB1 is a damage-associated molecular pattern marker, and it is released from the cell when the cell get exposed to or cell nucleus get damaged. And this is released into the cytoplasm. It becomes a very pro-inflammatory cytokine. And when we looked at our proteomic cargo from our PBDE exposed exosomes, we were able to show that HMGB1 is enriched in these exosomes coming from PBDE exposed amniotic epithelial cells. So we were interested because many of our oxidative stress inducers cause HMGB1 release. And that's what you are seeing here. Here is a normal amnion cells, and what is green is HMGB1, and what is red is exosomes in the cytoplasm. When you expose them with oxidative stress, such as cigarette smoke or PBDE, you can see the nuclear membrane gets damaged. The HMGB1 is released into the cytoplasm, and now it is in the cytoplasm as an inflammatory cytokine. It's just very inert and inactive, and it actually does a lot of DNA supporting function in the nucleus. But in the cytoplasm, it's a pro-inflammatory cytokine. You treat them with N-acetylcysteine or antioxidant, it is retained in the inside the nucleus. So this was exciting, and so I'm not going to get into, because of time limit, I won't get into too much of the mechanistic studies that we have done, but I'll show you these 4 papers that we have shown that HMGB1 gets packaged inside the exosomes, and these are exosomes from oxidative stress are enriched in these DAMs or damage-associated molecular patterns. And these exosomes can cause inflammation of the uterine cells as described in this paper. And if you inject these exosomes into pregnant animals, it can cause preterm birth. So there was some functional evidence that we can induce preterm birth by injecting exosomes containing these cargo. However, we need to show that in an experimental model in vitro that these maternal-fetal interface can actually propagate these exosomes from the amnion layer when they undergo senescence through these layers, through the chorion layer to the mother and activate the mother. So our initial experiments were using 2D cultures, which is a very limiting thing because you really do not replicate the intercellular interaction that is seen in these complex structure of fetal-maternal interface. So we decided to design an organ on chip. So one of my a PhD student who became my colleague now as a faculty, Dr. Lauren Richardson, actually designed organ on chip experiments. And we have published multiple papers showing that these organ on chip can be used to study fetal-maternal interface, human fetal-maternal interface in vitro in lab, and we can replicate the exact same phenomena that is happening in utero or in vivo in a lab chip. So what I'm going to show today is in the next few minutes, I will show this particular experiment, how we designed HMGB1 signaling using organ on chip. So this is a microfluidic device, which in this particular case, we have 4 concentric circles, and we can load cells. They're all interconnected through microchannels, about 300 micron or 600 micron distance. Each one of them are interconnected. And we can add media inlets, and we can take media or elutes outside using the outlets that we have built into these devices. So as this is such a small device, all these tubings can either add cells, add media, add your stimulants, and take the elute out when the experiment is done. So you can see this is a dye diffusion study, making sure that the media will not just diffuse between these chambers. And after that was done, we loaded them with 4 different cells: amniotic epithelial cells, mesenchymal cells of the extracellular matrix, chorion trophoblast cells, and the decidual cells of the mother. So we recreated the amniochorion structure here using these 4 chamber device, which also contain collagen that is coming from the human fetal membranes. We looked at the trichrome stain, making sure that nascent collagen production is intact. Also, the collagen that we added in the microchannels are also intact. The cells produced expected cytokeratin and vimentin markers, as you can see, and they were all showing viability up to 5 days in our culture system. So once we established that, we added exosomes containing HMGB1. For this, we engineered exosomes to contain HMGB1, and we electroporated them to contain recombinant HMGB1 inside these exosomes. We purified them, and after purification, we tested their particle size, which is found to be the same as control. And we wanted to make sure that electroporation did not damage our exosomes. So we did an electron microscopy study of the electroporated exosomes and non-electroporated exosomes that contain from normal culture cells. So we didn't see any change or any impact of electroporation on the cells. Similarly, exosome markers did not impact, were not impacted by electroporation. We also saw that our exosomes have HMGB1 as cargo, and they're attached to these markers. So we were successful in electroporating or engineering exosomes to contain HMGB1, which is a PBDE exposure, result of a PBDE exposure. So we were able to artificially create these exosomes. Now we added them to cells to make sure that, yes, the electroporated exosomes are indeed taken by these cells. Here is the maternal decidua. Here is the maternal myometrium or uterine cells. And you can see these green dots that are exosomes basically taken up by these cells. Regardless control or HMGB1 electroporation, they're taken up by these cells. And now we started our experiments where we added two types of, we actually added the electroporated exosomes, which is labeled with a green dye. And we also added amnion epithelial cells in the outermost circles, which contain RFE, because we also wanted to see whether normal exosomes migrate through these different layers and reach the maternal tissue from the fetal end to the maternal end. So we have the RFP containing amnion epithelial cells, and to the same chamber, we added electroporated HMGB1 containing green exosomes. So our goal is to see whether the red and green exosomes migrate from cell layer to cell layer to cell layer and causing inflammatory changes. As you can see, within 24 hours, they reach the next layer. Within 48 hours, they reach the third layer, which is still the fetal layer, chorion trophoblast layer. And within 72 hours, both red and green exosomes reach the maternal layer. So they can propagate from the fetal side to the maternal side, which is called communication between the fetus and the mother. And now I'm going to give you a graphic summary. I'm not going to get into the details because of time limitations. We were able to show that these exosomes can cause TLR activation because HMGB1 is a TLR mediated signal propagator. So TLR activation was seen in these cells, in the maternal cells. RAGE, another HMGB1 receptor, was increased in maternal cells. It also resulted in increased inflammatory cytokine production in the maternal decidual cells. And we wanted to functionally, physiologically show that this in vitro experiments can be replicated in vivo in an animal model. So we injected a mouse with time pregnant mice. On day 15 of their pregnancy, we injected these animals, different horns, with electroporated HMGB1 containing exosomes and looked for preterm birth. And as expected, these exosomes propagated from the fetal side to the maternal tissue and caused preterm birth before day 18. So we injected them on day 17. They delivered, all of them delivered before day 18.5. Normal delivery time is supposed to be 19 or 20 in this animal model. So in this particular experimental in vitro organ on chip model, we were able to replicate that in an animal model, suggesting that environmental exposure can cause fetal cell senescence, leading to premature birth. So here's a summary of our work. Whether it is physiological signals or pathological signals, cause P38 activation, senescence of human fetal membranes or fetal tissue senescence, causes damage-associated molecular pattern release, and cause NF-kappa B activation, inflammatory activation, leukocyte migration, And all of them further gets packaged inside the exosomes. And these signals are sent by the fetus to the mother saying that I'm ready to be delivered. It can happen physiologically in a normal birth. It can happen pathologically in premature birth. Now the mother starts to respond to the fetal communication or fetal signals. She increases her inflammatory load in her uterine tissue via NF-kappa B and eventually resulting in labor. I just wanted to give you one more example. We were able to inhibit this by giving NF-kappa B inhibitor as encoded inside the exosome. We injected an NF-kappa B inhibitor containing exosomes to the mother, actually in animal models. We were able to show that we can stop NF-kappa B activation, not only in the mother, but also in the fetus, because exosomes can pass through placenta, reach the fetus, and treat fetal inflammation and delay preterm delivery. So that paper is published earlier this year in Science Advances. If you're interested, you can read that paper and how we did this work. So to conclude my talk, congenitor-specific PBDE function can induce preterm birth. It can cause health issues in both mother and the neonates. Environmental toxins can generate ROS and induce cellular damage, fetal membrane senescence, as I showed, and that inflammation is propagated to the mother to cause preterm labor. So future direction is mechanistic impacts of environmental pollutants are hardly studied. And this is one of the very few studies that is reported the mechanisms of how it may happen. We need to look at this in all other endocrine disruptor work. Actually, my lab is now involved in looking at BPA and BPS and how that causes preterm birth. We already published a paper using the organ-on-chip on the impact of cadmium and another environmental pollutant. So this work needs to be funded. All these needs, many other researchers are involved in this research. We need more attention to the reproductive age women and their children who are suffering because of this. And I cannot be honored to talk today with the World Prematurity today to give this lecture and give this message. And I really want to thank Jerry and Hannah for giving me this opportunity. So this is, with that, I wanted to thank the funding agency. And without my lab, it's an incredible group of scientists and faculty and staff and fellows and students. And also my wonderful collaborator, who's a bioengineer who actually designs these organ-on-chip, Dr. Aram at Texas A&M University, and his students. There's incredible support for this work. And with that, I want to thank you again, and I'm happy to answer any questions.
Great, thank you so much. We're going to go on to the Q&A. I'm going to pass it off to you, Jerry. We're a bit over time, but we want to get to questions that we have so far, and feel free to put in a few more.
Okay, thank you very much. Wow, that was just mind-blowing. Starting with humans and going to cells and then to organ on a chip and back to animals and toxicants. It's really something. So I have a question. Well, like for these flame retardants and causing preterm birth, will that also work on miscarriage?
Absolutely. There are some evidence that they can also cause a miscarriage and stillbirth. So again, the mechanism is not well studied. Which congenitor PBDE is causing is not studied. So I'm not quite familiar mechanistically how it manifested, but definitely environmental pollutants, including PBDE, are associated with some incidents of miscarriages and stillbirth.
Okay. Another question. Are these fetal cells, do you think, more sensitive to flame retardants than regular cell cultures, perhaps?
It is. I do not know for sure, but this is probably one of the very few studies looked at fetal cells. Most of the studies are restricted to endometrium, which is and myometrium, which are mostly endocrine organs. And they're very well studied, but I think that fetal cells are much more susceptible because fetal cells are highly unlikely to get exposed to these because the placenta will act as a super barrier preventing them. But if they reach the intraamniotic cavity by challenging the placenta, by crossing the placenta, I think they are much more susceptible to these pollutants and toxicants.
Okay, last question, more of a general question. So what does the field in general, the preterm birth field, what do they think about endocrine disruptors playing an important role? Do you have a hard time working with them and convincing them that this is important?
No. Actually, there are quite a few. I have several colleagues and friends that are involved in endocrine disruption functions, and we always interact with them. And I think that the challenge is funding is not adequate enough for them to sustain their line of work. So that's one challenge. And the other thing is, experimental models. Animal models are not totally convincing to showcase that yes, that is what happens in humans. Endocrine functions are slightly different during pregnancy in animals. There are no close relatives to human pregnancy in any animals except non-human primates, but they are incredibly expensive to do.
Okay. I just have to ask one more question because
Sure.
I think it's important. When exosomes are released from cells, where do they normally go? Do they go around the body, and are they taken up by all cells, or do you think that they are headed towards a specific organ or tissue?
Jerry, excellent question. There's the tropism of exosome is still unclear. I mean, technically they get into the bloodstream and they can go anywhere. So when we look at them, one of the reasons that we really designed these organ on chip experiments, and we are actually expanding this from 4 chambers to multi-chamber, probably trying to include all the uterine tissue. So yes, technically they can go anywhere. And do they show any tropism if they cargo based on their cargo is still unclear.
Okay.
Yeah.
Well, thank you so much. That was
Sure.
absolutely terrific.
Sorry
I'll
if
turn
I
it back
take too
to
much
you,
time.
Hannah.
Yeah, thank you so much, Dr. Menon. It was a pleasure having you. What a great study, bunch of studies that you've shared.
Thank you.
All right. I want to thank you so much to Jerry. We're approaching the end of today's webinar. A video recording will be available on CHE's website soon, and tomorrow you'll receive an email containing a link to the video. The next CHE partnership webinar, Supplement Science and Regulatory Challenges, will take place tomorrow, November 18th. Then on December 1st, we will hold our next Young EDC Scientist Showcase Series webinar titled Potential Endocrine Disrupting Effects of Microplastics in Human Placenta and Aquatic Life. You can find details on our website at healthandenvironment.org. If you are new to CHE and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these CHE partnership webinars bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speaker, Dr. Menon, for taking time to present today, and to Jerry for your excellent moderation. Thank you so much for joining us.
Is there a way I can answer some of the questions in the Q&A? I'm happy to answer those questions. And if anybody wants to email me, that's fine as well. ram.menon@utmb.edu.
Great, and we'll go ahead and share that with our audience in our email that goes out tomorrow too, in case. Yeah.
Sorry, I may not have answered all the questions. So.
Yeah. Great, thank you so much, Dr. Menon.
Thank you.
All right, wishing everyone much health.
Bye.
Take
All right,
care.
thank you.
Bye-bye.
Bye.